I have to start with an apology because you will not find my curriculum and the synopsis of my talk in the brochure that you have, because there was so much to say about the bit that we decided at the last moment to divide the talk in two parts, and for reason of time I was not able to do it. But also the fact that my curriculum is not there, it doesn't mean that it doesn't exist. It's on the web and you can check it out. We have to do this because we have a different version of PowerPoint that doesn't work well. We have an version here, which it's an older version, and it should work. Okay, so I have called this presentation Ohmic Effect of Bridge-Induced Translocation. Omics, because as you know very well, the molecular geneticists and molecular biologists are very good at inventing new terminologies. I think this is due mostly to professors who are emeritus, and they don't know what to do once they are emeritus. I'm going to be emeritus pretty soon. So they sit there and they invent new terminology. So genomic, proteomic, transcriptomic, interactomic, atomic, and also omic. So what other effects there will be besides those that Valentina Tosato has just shown you about bridge-induced translocation? There are a few other omic effects. And so I will concentrate on a couple of these, which I've developed in the last 2 years. This project has been very, very successful. I think it makes me, in my last years of my career, it makes me feel good that I will leave with a good project that hopefully will be you know, food for further research in the years. But before doing this, I have to be a little bit the company man, and not to try to sell anything, but it was funny because this morning, you know, when the hospitality here is so good, and I have to really, I would like to do something. I would like to give a big hand to all the students who make this possible, because they are really very nice. This is something that I have proposed to do in Trieste with our students, because it's really a very good initiative. And it's so good that this morning, as I was sitting around here looking at the beauties of the city, somebody came and said, "Professor, do you need anything else?" can I get you anything? And as they do most of the time. And being, the usual Italian who cracks a joke, I said, yes, I'd like to have a Ferrari right here so that I can take a tour. Well, then there was a little discussion, we were doing Turkish, and then this guy moves and he goes inside and picks up the telephone. And then I said, no, it's a joke, please don't call me Ferrari. Because this guy was actually ordering a Ferrari to come here. Okay, so this is the place, just 5 minutes to tell you where we are from. I've been here before in the past years, but most of you are young students, so you don't know me. And this is the place. It's the International Center for Genetic Engineering and Biotechnology, which has the headquarters in Trieste. And what the students, because we should talk to the young people, the future, not the old one. We are down the share. This center is also yours. Why? Because it's an intergovernmental organization started by the United Nations in 1987, which is property of all the countries that participate in this organization, including Turkey. As a matter of fact, I'm here because I have first a master's student from Istanbul, and then a PhD student, and then I had Professor Ercan coming for 6 months in the last year. And so the interaction has started like this. And I invite all the students, because many of you have asked me, to check on our website, which is very easy. You just type ICGB and it comes out. It's icgb.org. Check what programs are available, because most of the programs are for students at various levels, at the undergraduate, let's say master, PhD, and postdoctoral level. So doctoral and postdoctoral level. Please do this because the institution is there and it's for you. As you see, this is the logo. We started, as I say, in 1982 as a special project of UNIDO, which is the United Nations Industrial Development Organization, and it was the first project ever in the history of the United Nations to establish a scientific institution with people from all over the world. This was the idea. In 1995, it became an independent international organization based on the countries that participated in it. So it's not even more a project of the United Nations. It's owned by the countries that participate. We have about 70 countries that participated. The mandate is to provide a center of excellence for research, training, genetic engineering, and biotechnology, advice to developing countries and economy in transition. Now, this last part has to be taken with a little with a grain of salt, because in '87 China was a developing country. Okay, now it's not a developing country anymore; it's an overdeveloped country, and so but it's still a member of ICGB and a WIPO member. But let's say that the mandate is to provide training for students from countries where either the conditions of the university is not allowing them to do exactly what they want to study, or just to provide a means to interact with other scientists and to enter a little bit the global arena of science. Okay. Having said that, I don't think I have anything else. No. Valentina has been discussing the results of bit translocation in yeast. She's made a quick reference to what Saccharomyces is. I would like just to go a little bit deeper to tell you exactly what this beautiful model cell that everybody's using. It's a fungus. It's categorized as a fungus. It's a single cell organism. Now it's becoming a model even for stem cells. Okay, why? Because it's totipotent. The cells can grow mitotically, like skin cells, or meiotically, like germinal cells, like ovary, oocytes or spermatocytes, and we can switch from mitotic to meiotic cells and back, manipulating the cell cycle at will with very simple means of giving or not giving certain nutrients, in particular fermentable carbon sources and nitrogen. It's a small cell. It's smaller than most mammalian cells. It's only 4 by 6, but it's still big enough to be seen at the electron microscope and to be used with all the techniques that are used for mammalian cells. In addition, you see it divides by body. So this is the mother cell, this is the daughter cell. And at the interface between mother and daughter, there's a neck, which is made mostly of chitin. And when the daughter separates from the mother, it leaves a scar on the mother. You see these rings of chitin, which are now used, counting them, to do aging studies in yeast. So yeast now is also a model to study aging of the cells. Because it's so simple now, you can count the scars that a mother has to know how old she is, okay? Or you can use special dyes that attach to these rings of chitin, for instance, carcofluor white, and then take the cells and pass them to the fax. Now, when you pass the cells to the fax, don't expect somebody else on the other line of the telephone gets the cells coming out of the fax machine. This is fax is called fluorescence activated cell sorter. It's a machine that you should know, some of you should know. It passes cells at very, very high speed through a nozzle, and it separates them into pools depending on the fluorescence. So basically, we can count at very high speed the age of cells in the population by this technique. And it's just one of the many, many applications. Also, I don't know if you know, but yeast has been the first eukaryotic cell ever sequenced by mankind. First eukaryotic cell ever sequenced by mankind. Its genome was released in Trieste in 1996 at the presence of Jim Watson and Craig Venter and all the other big shots of genes and DNA. And it's a small genome that it's a good model to mimic what happens in mammalian cells. Now, remember that one of you has asked the question, can this bit translocation be applied to mammalian cells? And the answer of Valentina was that actually it would be very difficult, because the big difference between this system, this genome, and the genome of mammalian cell is that this recombines spontaneously and also inducibly very well. Mammalian genome and mammalian DNA recombines very, very poorly. And this is the major drawback or the major difficulty, that all these technologies that are built in yeast now cannot be applied to mammalian cells. To make sure that also the students up there are listening, I will ask questions and give grades at the end of the lecture. Okay? The genome of yeast, as you see it, it's rather small. There's 16 chromosomes the haploid cells, 32 the diploid. It's only 12.5 million base pairs. Now, professors are always professors. You can't get away from this. So I will ask the question that I ask always to the students. How many of you, please raise their hand? I will spare the senior people, of course. How many of you, in there, honestly, know how much DNA do you have in terms of base pairs? Raise your hands, please. Just one. One? Anyone else knows how much DNA it has? Two? This is terrible. How many people, now let's stop this. How many people, raise your hands please, know their telephone number? Please, raise your hands. Please, raise your hands. All of you, come on, all of you know your telephone number. Okay? You have two telephone numbers, but you're a professional. So this is terrible. everybody knows his telephone number, but almost nobody knows how much DNA he has in his cells. It's an experiment I always do, with students. Now, let's see. The one that who raised this hand, how much DNA do we have? 3 billion. So 3.3 billion. Now you know that you have, I inform you officially, that you have 3.3, roughly, billion base pairs in your DNA, in your genome. Please remember this because it's more important than your telephone number. The telephone number you can change it tomorrow, but the number of base pairs that you have in your genome, you cannot change it tomorrow. Okay, so let's continue with this. The genome of yeast is like all eukaryotic genome. It's made of individual chromosomes. They are small because the overall is small. It goes from 200,000 base pairs to 1.5 billion. It has, the genes are organized like all the eukaryotic genes, promoter, overtrain, terminator. The genome is very compact. By sequencing, I had the privilege and the honor to be one of the 100 laboratories around the world that sequenced the yeast genome, besides having the great honor to host the final genome conference in Trieste in 1996. We learned many things about yeast, but about genomes in general. The first was that the smaller genomes are, doesn't mean that the number of genes decreases linearly. The bigger the genome are, it doesn't mean that the number of genes increases linearly. As a matter of fact, years ago, the human genome was supposed to have 100, 150,000, depending on the speaker. If it was a bigger headed, it was big number of your genome of man is probably 375,000 genes. Okay, it's not true. We now know that the genome, how many genes do you have? You have 5 genes? Bravo, bravo. What is your name? It doesn't want to say. Anyway, his answer is correct. We have about 30,000 genes, some people between 25 and 30,000, so much less than we thought, because our genome is more than 200 times the number of genes of yeast. Yeast has 6,000 genes. When it was sequenced, the paper in Nature was entitled Life with 6,000 Genes. Now, if the genome of man is 200 times bigger, then it should have 6 times 200, it should have 1,000, no, it should have 1,200,000 genes. It's not true. But, rightly so, recently, you know that there's been a great work program called, what was called? ENCODE. No, it was not the genome project. It was called ENCODE, Encyclopedia of the Gene Definition. And this project discovered that what we call junk DNA in humans, indeed, is made of many switches. Okay, so you know that there was always the story that small organisms have the genome very compact, just genes and nothing else. Big genomes have genes, but then a lot of junk DNA. We used to call it junk DNA. Now we know that junk DNA is not junk DNA. It contains 6 million switches on and off that interact with each other to trigger the expression of these 25,000 genes. So, to say this is a compact genome, it's much better to study genomics in a small organism than in a big one, because otherwise it becomes very difficult, of course. The other nice thing about yeast is that you can do this. This is a phosphate gel electrophoresis, which separates all the chromosomes of yeast. all these bands are individual chromosomes. And it's very hard to do this with mammalian chromosome, because mammalian chromosomes are huge, 100 million base pairs each. And this is difficult to do. You can do this very easily in yeast. So yeast is being taken as a good model for the human genome, taking into consideration what we said before, that recombination in human cells is so inefficient that most of the genetic engineering tricks that we can do in yeast, we cannot still extrapolate them to mammalian cells. Okay, so let's go back to the what other omics, what other effects this bridge-induced translocation has demonstrated. You've seen this before, and as you remember, this is basically a profile, expression profile of genes around the point of translocation. So this is the bridge, the cassette that leads to different chromosomes, and around the point of translocation. Here you have this wave of overtranscription, and it happens every time we have a translocation. So it seems that when you change the natural configuration of the DNA, okay, because this is used to be a unique piece of DNA. Now this is linked with this, which is not what it was before. Anytime you do this, you have this wave of alteration of gene expression. But how big is this wave, and how extended is this phenomenon? By doing expression analysis of genes on both sides of the translocation point, on one side or another, near or far away, and then transforming these, I'm sorry for this, this is what happened by converting, but you can see the graph. Transforming those data in histogram, you see that genes, these are genes which are on one side or other side of one breakpoint. Only this, which is 50 kilobases, it's only 1.5, 1.3, 1.4 times higher. So it looks like that if you go far away from the translocation point, this effect stops. Okay? And it's only localized. Then at this point, to understand exactly what was going on the whole genome, to see how many of these genes were indeed activated by a single translocation event, we have to go on microarray. And I would like to draw your attention, if it's going onto this spot. You see everybody, right? So you see this spot here. This is the most important one. And you believe me. You have to take this, as a whole. It's a green-red expression profiling, meaning that if the green profile and the red profile, the wild type and the translocan, have the same intensity, they will give yellow, because the red and the green in the same spot will give yellow if they are in the same quantity. If it's underexpressed, it will give red. If it's overexpressed, it will give green. Well, one would expect that if the big translocation did very little on the whole, this is 6,000 genes, repeated many times, we should see all of this yellow, maybe some spots here and there. But instead, you see how many red and how many greens? You see this? One single translocation, Dmitry Nikitin, my Russian postdoc, sat down there and really did a very thorough statistics, which I will discuss a little bit with you later. On his way to do proteomics, he calculated that a single translocation event affects more than 1/6 of all the genome. So over 1,000 genes. A single translocation event. This is why the cells that Valentina has shown you, they have lost the cell cycle control, they have lost nuclear localization, they have so many different monstrosities that indeed look very much like cancer cells.
When we published that paper in PNAS, and we concluded by saying that this could be a valid model to study cancer cells in yeast, then the editors, I hope nobody of the editors will see here. One of the editors said, well, actually, that's too much to say, and you have to demonstrate other things. Well, little by little, we are demonstrating that indeed most of the things that go wrong in cancer cells can be found in, analogously in big translocations. So if we take all this data, we compound them, chromosome by chromosome, we see something like this, for example. In a translocation, this is a different translocation. In a translocation between 8 and chromosome 15, okay? This is where the cassette is, this is where the translocation begins, and you see that this region, up to the telomeres, changes, switches expression. The chromosome means the average expression of the gene. And you can see that indeed on the right arm of the chromosome, some are up, some are down, but basically that's the baseline. Here you can see that all of them are higher. The same goes here. Okay, this is the translocation breakpoint. This region is all higher than normal, and then it stops at the center. So our current interpretation is that we don't know why. I mean, that's the truth. We don't know why. We are guessing. It's probably chromatin. It's probably an effector, a transacting effector. We don't know why. But every time that we produce a chromosomal translocation, that is a dramatic event. In humans, as you heard, it produces cancer. Many leukemias are produced by translocation. The first one, the most famous, is the Philadelphia chromosome, right? It's the connection of two different chromosomes. Well, every time we destroy the normal organization of the yeast genome with a translocation, we generate a wave of overexpression of genes that it's stopped on by telomeres or by centromeres. Okay? And if we now consider, follow the laser beam, that the new translocal chromosome is this, okay? Then in this new translocal chromosome, the entire left arm is overexpressed. Because now this will be part of this, so this will be overexpressed from the centromere to the telomere of the new translocation chromosome. A huge effect on translocation, on gene expression. Obviously, after having done gene expression, I will go very quickly now, the next thing to do was to do proteomics, because having the genes that are highly expressed doesn't mean that the proteins are also highly expressed. There is a step in between, and we did basically a proteomic analysis by the usual way, very laborious. This is the wild type. These are two translocans. These are the 2D gel electrophoresis, in which the proteins were separated by weight and by isoelectric focusing. And when you do these experiments and you have to repeat it many times, it becomes really a question of interpretation, as the cartoon says. But this has to be repeated many times to make sure that you have good reproducibility, that all of them are combined in a single one. This is the wild type, remember? Then the two mutants are compared to the wild type, and the computer goes and picks up the difference in spots. And all of this is done by computer. So the computer circles all the spots that are either underrepresented or overrepresented. Based on this analysis, this is how proteomics is done in the non-robotic way. There is also a robotic way, which is like this. You put the cells here, you push the button, and then you go to have a cup of coffee, and you come back and there are the results. Since I like to do the experiment the old-fashioned way, this is the results that we have obtained. All the circled spots are the ones that indicate proteins either over or underexpressed. all this data were collected, and the proteins were categorized in various categories based on their profiles: structural, DNA repair, cell cycle, stress response, other proteins, and so forth and so on. And in the end, Filippo Macassoli, who was the graduate student who did most of the work, working with my postdoc Dimitri Nikiti, produced this nice pie, which was the result of his work. And it was a big surprise. Why? Because we were expecting, dealing with recombination, dealing with DNA replication, dealing with, let's say, DNA metabolism, we would expect that most of the genes affected, whose transcription would be affected, were DNA metabolism genes. We were wrong. We were wrong. Remember, students, this. Most of the times, a famous Nobel Prize at Southern, responsible for the Southern hybridization, once said, experiments usually don't work. Okay? So remember this. We thought finding a big change in DNA, DNA repair is the smallest category of genes affected. Now, one could say, well, because you have less DNA recombination genes with respect, but these are proportional. In other words, 3% of all the DNA repair genes are affected. Okay, not 3% of the whole cell. Whereas half of the metabolism genes are affected. And this was a big thing to think about. The nice thing about our job is, I was telling some of the students today, in our job you don't make a lot of money. I mean, let's be honest, okay? And one of the students said, oh, that's exactly what my dad says, you know, always. You don't make a lot of money, but sometimes you have interesting, funny times. I find most interesting times when I have to sit down and try to interpret the data. I like this. I like this because you have to use your brain, okay? And it has to correspond to something that you can demonstrate. Why? The greatest category of genes affected by translocation, chromosome translocation, should be metabolism. 19% stress response, it makes sense because you stress the cell a lot by doing this event. Cell cycle, we have seen, it's DNA synthesis dependent. Structural, well, in some sense, you change the structure of chromosomes, so structural genes should also be interesting. Unknown, that nobody knows, but why the great majority of the genes are metabolic genes? The only thing that we could come up with was this thinking, that if we accept the Darwinian principle of evolution by mutation and selection of the fittest, we can appreciate how this event, how this system is slow in promoting adaptation. And there are now, if you read the literature, a lot of papers coming out on adaptation, which tend to go in this direction. To quickly adapt from a healthy, plenty of food, plenty of water, with nice sun environment, to a desert environment, without being wiped out by selection, some dramatic event must occur. Single point mutation would not be enough to take Arabidopsis thaliana, which is a plant that grows in the bushes. It likes water, it likes sugar, it likes, you know, the table full of food, and become Arabidopsis arenaria, which lives on sand. You cannot do this very simply, very quickly by single point mutation. There must be other events that have an evolutionary force. Gross chromosomal rearrangements, it's one of these. And chromosome translocation, we believe, is the strongest, still life-compatible genomic rearrangement that can change dramatically the metabolism of one cell, so that immediately from a rich environment you can adapt to a dry, harsh environment. It's a hypothesis, but it will explain why the majority of the genes affected are metabolic genes. Okay, we have seen this, so I just can move on. Gene goes to the translocation point. Also, the proteins, not just the genes, are highly expressed. This is what it means. Okay, so the conclusions from this proteomic. Translocation event leads to changes in the level of protein expression. This differential profile belongs to most major functional categories. Gene close to the translocation point seem to be affected by effect. And then the metabolism seems to be increasing, particularly in some mutants. And so the cell cycle machinery changes to reflect some delay in proliferation because the cells goes a little bit slowly. And here is some conclusion. Maybe it's the cytoskeleton of the cell that is affected. Then I would like to conclude with one other astonishing discovery that we made, that was mentioned by Valentina but was not described in detail. She said that one of the big problems that arises after one translocation event is that you have multiple rearrangements occurring throughout the genome. So one translocation in a particular location affects the whole genome. And this work was done by Beatrice Rossi and other students in the lab. Basically, a translocation here you see between two SUSU1 and SUP2, two genes and other chromosomes. This is chromosome 16 and this is chromosome 9. Generate a variety of, besides the translocation chromosome, a variety of arrangements. And these arrangements, this is the control strain, show also different phenotypes. So different phenotypes, this is an aberrant phenotype, is the cell budding and becoming hyphal instead of being single cell. Here you have the partitioning of the DNA within the same nucleus, multiple nuclei, as you see also here. So there are several defects that we can see cytologically. And we were able to discover 10 different categories of mutants that are generated by a single translocation. So a single translocation, as far as we know, can generate up to 10 different gross chromosomal rearrangements, each resulting in a well-defined phenotype. Here the phenotypes are analyzed also by fluorescence. This is the same staining that Valentina has shown. It's called FUN. And it relies on the detection of these proteinaceous bodies in the cytoplasmic vacuoles when the cells are normal. Then when the cells begin to get sick, the vacuole degenerates, this material invades the cells, and finally, when the cells become yellow, they are dead. And you can see different percentages of death in these 10 different genomic categories. Then we looked at copy numbers of genes. These are just the two genes involved in translocation. And we basically came out with the conclusion that out of a single translocation, 10 strains, 10 different strains, from SUSU1 to 10, having different number of genes in different parts of the chromosomes, were produced by a single chromosomal translocation. Okay, just going a little bit too fast. As a matter of fact, when we do hybridization with different probes on different fragments of chromosomes, we see different genes segregated differently. In this case, for instance, we have 4 copies of the same gene, which means that the cells has become a tetraploid. Polyploidy is therefore generated as well by chromosomal translocation. I move forward. These are the final 10 genotypes that we discovered from a single translocation, and therefore published in Genetics, demonstrating that the cells adapt immediately to a translocation event by rearranging the genome in many different ways, in as many ways as it can. Okay? And we again are at the point where we have to understand exactly what is that out of a translocation triggers this power in the cell. I want to conclude with to render homage to my colleague, dear friend, Erjan Arikan, who's not here because he has a family problem, unfortunately, who has been with us for 6 months. He was very interested in bioinformatics, and so I gave him a project when he came, starting from the Italian stereotype. What is the Italian stereotype? When you see an Italian, usually, unfortunately, what is it that you think? Spaghetti? Bravo. See? He studied a lot. Bravo. Spaghetti? Give me another stereotype. Huh? Pizza? Milan? Once we had this famous stereotype of lovers, big lovers, but I think it's going down now, it's changing. But the most important one is always spaghetti. If you see an Italian, they take spaghetti. And you better think, because they're very good. Okay? I did my postdoc at the University of California, Berkeley, and I had the privilege of having as a mentor Professor Seymour Fogel, who founded the genetics department. A fantastic man, responsible for many studies on meiosis and recombination. I remember that he used to say, when I die, I want this written on my tongue. He loved pasta. Okay, so because of this, one day this is another stereotype. All the Italians are comedians. I'm teaching a course in genomics at the University of Salzburg every year, and because I also like Mozart, so I have to go to the theater. And I was there in January, February, and I had to explain to the students the principle of DNA structure. And it's difficult because we have to use a two-dimensional meaning tool to explain a three-dimensional problem, right? So either you come with a statement that shows the DNA structure, and I was thinking, how can I make the students understand it? And that evening I went home, and there was, of course, pasta, with pasta col sugo. And as I was eating, I looked at this. Oh, it looks like DNA strange. And then I made some DNA model with spaghetti on the table. As you can see, this is a little bit, a little bit of juice, which is that. I had to explain the difference, a very important difference, between two fundamental structures of DNA, bDNA and zDNA. Why is this? Because bDNA, as you know, is the usual right-handed double-stranded molecule that we find in solution in the nucleus of the cells, compacted with chromatin. All of this is chromatin. You see that it falls apart depending on the prescription of the genes. Okay? But from time to time, the DNA can assume other configuration. The most dramatic of which is zDNA, which is left-handed. And I have to explain to the students what happens going from right-handed to left-handed. I studied this when I was at Berkeley, so I remember I made a model which came from looking at the fence you know, these fences that they make with wire, you know, because I have 2 dogs, I have this fence with wire. And one day the dog ate a piece of the fence, so I beat him up, and then I picked up the fence and I looked. And to make the fences with wires, the companies, they go like this, and then all of a sudden they go the other way to make a hole. In this hole, it goes the other wire perpendicular. Then it was done with spaghetti. It's this. This is the right-handed DNA. Okay? This is the right-handed DNA, which categorizes the B structure. This is the left-handed DNA, which categorizes the Z structure. You see what happens when you go from B to Z? At the transition, you open the 2 strands. And when the DNA is open, it's a fantastic site. It's an invitation for proteases, recombinases, transcription factor, promoters. Yeah, promoters actually, they open up with a system which is similar like this. So the B to Z transition is a very important point because it makes the DNA more recombinogenic. Then I had when Ercan was there, I said, why don't we do this? Why don't we look at the primers that we use to make bit translocation and see whether or not there is a zDNA inside by computer analysis? Because if there is a zDNA inside, it means that the more open structures you have, the easier it is to have chromosomal translocation. And this was the project of Ercan, who unfortunately is not here to listen to me. But he did a very nice job, and he left Trieste with a nice manuscript, which we are now submitting. So basically, this is the guy, and he started to look for computer servers which do this type of analysis, and look for zDNA motifs. You see, for instance, this is a typical zDNA motif which is found in one of the regions that we use for translocation. Every time that you have a string of alternating purine and pyrimidines, you have a change from right-handed to left-handed. At the interface, you generate a hole, and that hole can be the site for recombination. Ercan did the analysis, all the computer analysis, and this was showing some of the results. Here they are. Some of the results. So he found many of these regions. Okay, this is the, of course, the significance. Okay, so the more, as you know, with the Lippmann-Pearson algorithm, the more negative it is, the more significant it is. So, which means that this primer on this chromosome that we use for translocation does contain a bona fide B to Z transition. In the end, we could conclude that in these regions where we generate translocation, not only the length of the homology is important, but also the presence of zDNA. The presence of a zDNA structure increases the probability of recombination and therefore the probability of chromosome translocation. And this is what the story of BIT is at this point. And I thank you very much for your attention. And this is the Gulf of Trieste during the largest sail race in the world. It's called Barcolana. It occurs every fall during the month of October. Over 2,000 ships come to compete with each other. Questions? Yes. En français, hein?
So, yes, don't worry. Yes, just before, you mentioned that one classification leads to about the alteration of expression of about 100,000 genes.
No, 1,000.
1,000, yes. And how many proteins have you find? how many changes? What is the correlation? Is there any? Yeah. Maybe there is no correlation. That
no, I got the question. So in other words, how many of these operational genes result in
seen at
about 90%?
90%.
90%.
90%.
Thank you. Yeah, it's a very high percent.
Because I was clear on the detail. You know.
Thank you. Yes.
So the question.
At the beginning, you mentioned that yeast genome alterations cannot applicable for human cells. But I missed that point. Could you please explain again that part?
Why it's difficult to apply these technologies to human cells? We are trying now, we have a project in which we are trying to eliminate chromosome 21 from a line of fibroblasts, mouse fibroblasts that has a human chromosome 21 additional, to see whether or not it would be in a future, it would be possible to tackle, for instance, Down syndrome. Why is it difficult? Because every time that you have to recombine, that you have to make recombinant DNA, that you have to insert or knock out the gene, or modify a promoter, or change the sequence on a mammalian cell, it is extremely inefficient. Extremely inefficient, because the homologous recombination in a mammalian cell doesn't work well. The major pathway that works in mammalian cell is called non-homologous end joining, which means that if you have a double-strand break, any two ends can be put together, not just the one that you want. Whereas in yeast, homologous recombination, which is very specific, works much better. So in the human cell, homologous recombination is inefficient, and therefore that makes it very difficult to manipulate the mammalian gene. Other questions?
We can get the other question in the break. Is it okay? We can get the other question in the break.
One last one, because he guessed nicely. So the spaghetti man has the right to have a question.
This is a very quick question. What controls the gene, actually? The regulation of the gene? In a sense, simple that what controls the gene expression? I mean,
that goes up and down. Yeah. Why are the genes going up and down? I could tell you a lot of stories, but the real answer is that we don't know yet. We have some evidence that the chromatin structure becomes looser, and therefore the accessibility from polymerase and transcription factor is higher. But we did that only on one gene, and therefore I cannot say that it's the general. We have recently seen also that general structure, like the cytoskeleton, which also has to do with compacting chromatin, is affected, and this could be a way to control the expression of these genes. But it's only guess. The real answer is we don't know yet.
Thank you for your presentation. Now we will present your certificate of 2 saplings planted in your name in the 7 Trees Forest. Sayın Doktor Carla Purisi'ye çok teşekkür ediyor ve adına dikmediğimiz fidanları sertifikasını Ve şimdi ikramlarımız devam ederken İstanbul Üniversitesi'nin en iyi müzik grubu seçilen Angarya grubu canlı performansıyla birazdan karşımızda olacaktır. Tüm katılımcılarımız davetlidir. İyi eğlenceler.